Hi, I am the research coordinator for one of the studies that Barbara is doing, and I want to thank you for allowing me to horn in and make a little spiel for this study. All of the gut mucosal studies that have been done have pretty much been done in men so far. With Ruth Greenblatt, who is the principal investigator of the Women's Interagency HIV study, which is the longest running natural history study of women in HIV in the United States. We are opening a study where we will study gut mucosa and endometrial mucosa. We will be doing colonoscopies, not sigmoidoscopies, so people will be sedated, so it will not be torture. There may be a little bit of mini torture the night before because you do need to do a bowel prep in which you need to take a very strong laxative. But all of the procedures that we do will be under sedation. We will take 8 to 20 colon biopsies and also some endometrial biopsies. Obviously, it's open only to women. It's open only to women who are still cycling, meaning having regular menstrual periods, and women who are not on any hormone therapy. It's a really exciting study because there are a lot of similarities between gut tissue and endometrial tissue. The biggest one being immune tolerance. The gut tissue really needs to be able to distinguish between foreign antigens in food. It needs to be tolerant of bacteria that is normally in the gut. And the endometrium, which is the lining of the uterus, if you didn't know that, sorry, needs to be tolerant of fetus. And so it's really a very exciting study, you know, that we've studied the gut, we've studied the female reproductive tract, but never together. So if anybody is interested in doing this study, there are consents in the back, and I will be here till at least 2:30. We're also doing another study about women and menopause, where we are asking women just to have cervical biopsies and some cervical cells collected, no endometrial biopsy. And that is actually also another really important study about reproductive tract mucosal immunity because, as we know, the population of people with HIV is aging. So that's it. Thank you so much for letting me horn in, and I will be here if anybody wants to talk to me about any of these studies. Thanks.
All right, well, I'll second what Barbara says because since I am the last speaker, I will go quickly through some of the slides that you already know. We're very fortunate in having a group of long-term survivors. We called them that when we first started studying them in the 80s. And we interact with everyone that's here, which is really nice. And some of my subjects are here in the audience. They've been fantastic. I have people that have been coming to us since 1982. And we have people who are long-term survivors, which we like calling them. I thought it was a little more positive in the 80s, who have been infected more than 30 years because they were in the hepatitis study. So takes back to 78, perfectly healthy, CD4's 800, and they're doing terrific. And as I say, many of them go to the NIH, many of them go to Boston. Steve and I and Peter work with them, so they're shared. And I think one of the things that I hope comes from this meeting, and I congratulate Lorene for putting it together, is a lot of collaboration among the groups so that we can share the information. All right, so there's the virus. And here it goes budding from the cell surface. I put together some animation to kind of relax the group, and I'm glad it's the last talk, so you'll just see it replicates. And when it replicates, it goes through up to 10 mutations each replicating cycle. But the key is, as it replicates, it becomes more cytopathic and can go to various tissues, including the brain and the gut and so forth. So it's a tough job to control it, and there are a lot of cells that are susceptible. Everyone talks about the hematopoietic or the bone marrow cells, but the skin, the gut, the brain, the prostate, a lot of cells are infected. And the other important thing for everyone to remember in basics is that this time we're dealing with an infection that's not just a single virus like chickenpox or measles or mumps. This is infection with the virus-infected cell as well. In fact, I think that is the major culprit. It then can interact with a variety of cells in the immune system or the gut or in the mucosal lining cells that Barbara talked about. This is, for example, seminal fluid. We did these studies many years ago looking at a lot of seminal fluid by in situ hybridization. These are 2 infected cells in seminal fluid. There's about 1 million peripheral white cells in seminal fluid normally, and that would be, if you do the math, 50,000 infected cells. So you can see why this type of infection could really lead to transmission. And if we looked at the same amount of infectious virus, it's somewhere between 40 and 50. I still get controversy about this. There are some really good studies now supporting the fact that infected cells are a major culprit. This is a great picture by Dave Phillips showing an infected macrophage interacting with cervical epithelial cells. You cannot infect those cells directly with virus, but you put infected cells on that cervical tissue, it infects, and David has time-lapse photography which shows that cell marching from one epithelial cell to the other, delivering virus. So this is a major way of infection. Now I thought it'd be fun, I use this for students, so this is how the HIV replicates in cells. Just get oriented. There's HIV, there's viral RNA, becomes DNA, and then you form the core proteins, the inner core and the envelope. So the virus interacts with CD4 and co-receptors. The core is put in. One of the RNAs is duplicated into a DNA. That DNA duplicates itself and forms a double helix like the chromosome. It then goes into the cell and integrates into the chromosome of the cell. From that proviral copy, it makes RNA. RNA makes all the proteins. The proteins assemble at the cell surface, and it buds from the cell, carrying the yellow spikes, which are the envelope. You can watch it again. So this is what we're dealing with. We're dealing with control of virus-infected cells. This is very similar to cancer, very different from other types of infections, because in order to control, you've got to take care of this, not just the free virus. So our definition is very similar. I'll just say that, and I'm pleased to see now that most people are calling elite controllers at least 10 years. It's remarkable, very low, undetectable virus for that long period of time. But my own impression is this is part of the bell-shaped curve. They're at the far left, very good control, long-term survivors at the right. I really don't expect any big differences between the two. You have innate and adaptive immunity. I'll just say that the immune system, you can get a little picture of this. Again, these are from my student talks. The immune system goes, if it goes through the thymus, you get the cell-mediated type of immunity, where there are T cells that you've heard about, they're formed. And if you go through the bone marrow, you form B cells forming antibodies. And the innate immune system, which has been around for millions and millions of years, has a large number of different cell types on the left and soluble factors on the right. This gives you an idea of NK cells, which we heard a little bit about with Doug's talk, where they will destroy virus-infected cells. Adaptive immune are the ones that come later, and those are the cytotoxic type of T cells, CD4s that help, and B lymphocytes, and there's some pictures of them by electron microscopy. This I always like. This is the macrophage, which plays a fulcrum. It can be in the innate or the adaptive. It's putting out its pseudopod to pick up that bacterium. It then takes it in, like macro, large phage eating, and then expresses, as some of you have seen, the proteins or the small particles of that bacterium on its surface and begins the recognition of an incoming pathogen. So the innate immune response, the earliest these derives to various cellular factors, cytokines, that help in both adaptive and in innate immune responses. Now, I had to put this in. Tony Fauci sent this to me a while ago. Cytokines are very important. You've heard those. Those are cellular factors I mentioned. It is, they all interact. If we try to interact, if we try to disturb one of them, we can get quite a disruption of what goes on. I was up in Davis a long time ago, and Murray Gardner, whom some of you know, said to me, "Gosh, Jay, that looks like the Paris Metro." So I always like, and I used it at our meeting in September 25th because Francoise was in, but I had to say it's true, it does, but the Paris map is easier to decipher. So we work with two types of cells, and we really evolved into an innate immune laboratory. I'm really a trained virologist, a medical virologist, but when I learned from long-term survivors the immune system's responsible, we've quickly moved into immunity and worked with CD8 cells, which I'll tell you about, but essentially then realized we're working with the first level of defense against HIV, which is the innate immune system. So it comes within minutes to hours. And plasmacytoid dendritic cells, which are the producers of interferon, are part of that. And what you're seeing here in the second red bar is if you take an infected CD4 cell and interact it with this plasmacytoid dendritic cell, the interferon is produced and you bring down HIV replication. So it's a very important antiviral cell in the body, and we have found that our long-term survivors have even higher numbers of these cells in healthy donors, and then as you progress to age, you lose them. What I think is important in looking at the role of this cell is we have an acute infection program called the Options Project, which we ran for 10 years with Rick Heck and others. And when people first come in with infection, we measure the PDCs, plasmacytoid numbers, and the viral load, and you'll see that if the person has high interferon-producing cells, they usually come in with a low viral load. Now, what's happened? They come in with a low viral load. 5 minutes? Have I talked for 7 already? Then they will have we think these will go on to long-term survivors. All right, so what happens here then is that the infected the PDC goes into the lymph node where it interacts with infected CD4 cells. That releases interferon. It produces, the PDC becomes a mature dendritic cell. It then acts in the adaptive immune system to elicit T cell responses. Okay, so I'll get through this and go to the non-cytotoxic response of CD8 cells. And this began with an individual we saw in '84 who was perfectly asymptomatic. The first report of a carrier, and then we found virus in his blood, and then after that we couldn't isolate virus all the way up to the present time. And what we found is if we took the CD8 cells as shown here from the blood, the virus came out. So it said, and you'll notice the CD4s are still there, so that meant that these CD8s were not killing, or else you would have eliminated the virus-infected cells. To prove that, we took virus-infected CD4 cells, added in the CD8 cells again, and what you see is you suppress virus replication. So this now became what we call the CD8 non-cytotoxic antiviral response, which you see on the right is a suppression of virus replication, whereas on the left is what you've been hearing about, more of an adaptive immune response where you involve cell killing. What we're finding is that the innate type of response, suppressing activity, is a major way of controlling the virus, and we think is an important component of long-term survival. If we separate the CD8 cells from CD4 cells, we will find that the CD8 cells secrete a soluble factor that suppresses virus replication in CD4 cells. That factor is called CD8 antiviral factor. Some of you here who volunteer for our studies know this is really key. We need people who get control because we need lots of fluid from CD8 cells to isolate this factor. And I'm going to leave some brochures in the back if you're willing to take part in this. It's really very helpful because as far as we're concerned, if we can find CAF, we will have a major mechanism for controlling this infection. So if time is really laid out, then maybe I'll stop here and you can move on to the next subject. We can have questions.